Thank you so much. I appreciate the opportunity, and we'll be happy to present our data. Um, I have been in research for a long time, and we focused on melanoma because we wanted to develop a translational research program. That was important for us to always have the connection to patients' needs, and then build basically a street from the bed to the laboratory, as well as then back to the clinics. And so let me give you initially a brief introduction of the teams in my laboratory. We are a signaling inhibitor group, but then also we got into immunology simply because we were unsatisfied with our current models. And I may point out to some of the folks who have been involved in this research. So let me give you a brief overview of where we have come from over the last few decades. And there's the classical 2D approach. Then more recently, the spheroids and organoids. I will not go much into details of this work. We then worked also on immune models that were connected to reconstructing the normal human skin, and finally, a variety of mouse models. And then, of course, in recent years, we tried to get as much information out of this, out of the cells and that is our major drive, try to model the cells as closely to the human situation as possible. And we have done that mostly or almost exclusively through NIH funding over the years, and that is going on. I did have some industry funding, and then after a few years, I didn't like it anymore. So we changed back to purely an NIH-driven research program. Let me just very briefly give you the overview of our PDX program. And we modeled it a little bit after the TCGA, because we wanted to have enough specimens to deal with, also for omics characterization, and to have a strong representation of all the clinical, genetic, and biological groups. So the tumors were implanted into NSG mice, which we got from Jackson. And then there was one sort of special case where we were early on very interested in drug-resistant tumors and what can we do about drug resistance. So the mice were fed with the drugs if the tumors came from relapsed patients after treatment with kinase inhibitors. And so these cells, we call them resistant PDX, and then these cells then were undergoing extensive genomic characterization. I just summarized it very briefly here to give you an overview, because melanoma is a high mutation burden tumor, which the immunologist welcomed because they are more immunogenic. However, the biologists have a nightmare to deal with them. So we can say today that we have, through these 500 PDX, from this one tumor, we basically have all represented groups. My own laboratory did the initial growth and the characterization, for example, of metastasis, and then the characterization. We are almost there, meaning I estimate by July we should have all the whole exome sequencing, all the RNA-seq, and then we are using a platform from the MD Anderson on targeted protein arrays called RPPA, reverse phase protein arrays, to give us at least the first glimpse of what is happening at the protein expression levels. And so that was a major, and I don't think it's a very smart way for a academic research lab to make that many PDX. But we are now in the process of having some major expansion of it. And this work is right now very acute in the laboratory, where we are looking for mutations in 10 pathways. And we selected it from a recent paper by a Memorial Sloan Kettering Cancer Center group in Cell in 2018. And as you can see here, 94% of melanomas, that is from the TCGA data, have, or 94% of receptor tyrosine kinases, 36 receptor tyrosine kinases this group has included, they had mutations. And you can go down the line, the cell cycle, of course, the PI3 kinase, of course, the P53, but then we hadn't paid much attention to Notch, Wind, Hippo, so these classical developmental pathways that are mutated in melanoma in about a fourth or 1/3 of the cases. And this, I believe, is also important for our understanding in the immune response and certainly in the growth property of cells. We have very similar numbers in our PDX. We just completed the first 250 and the others will be added. So what was in the patient, we find generally in the PDX. And we have actually in Jeff Trung, who is part of the PDX net and heads the coordinating center for this consortium, has also shown extensively that the genetic operations in PDX and tumors are relatively similar. They are not the same, but similar, and we have some shifts, but nothing really of major significance. So these type of genetic analysis, together with the gene expression analysis, will also guide our therapy in the next few years, because now with the PDX, we can validate this. Before I get into the immunological questions, let me just present 3 challenges for the field. And they may or may not be specific for melanoma, but that is a sort of a prime model for it. And the first is that single tumor cells that you get out of patients and you stick them in an NSG mouse and you're patient enough to wait for a couple of months, they will start to grow. So and this is basically killed for us a cancer stem cell model that there are 2 different populations. Every melanoma cell, and this has initially been shown by Sean Morrison, who is now at UT Southwestern, every melanoma cell is considered to be tumorigenic and can do all the things a melanoma cell can do. That's a big challenge, and it's a real challenge for the future when it comes to therapy and how we can eliminate these small populations that we call the drug-tolerant persisters. So while we have said there are no cancer stem cells in melanoma because every melanoma cell is a cancer stem cell, we still say there are subpopulations with stem cell-like properties, and I will come back to that in a minute. The other challenge is that melanoma cells are stealthy. They hide. So melanoma are derived from melanocytes, and melanocytes are neural crest cells. So when you look at melanoma cells, you find all the fibroblast markers, almost all the properties of a fibroblast, and one may call it the mesenchymal mimicry. So the enzymes, the growth factors, the way they put down the matrix, it's almost a classical wound healing response that you see in tumors, and Hal Dvorak said this some 20, 30 years ago. And it's not so dissimilar with actually endothelial cells, where Mary Hendricks at one time called it also the endothelial cell mimicry. And melanoma cells, if the need arises, they can make primitive vessels. And that was my estimate, 70, 80% of, and even endothelial markers are also expressed on melanoma cells. For the fibroblast, if you look at individual markers, you find them all on melanoma cells. However, if you use some combinations, you may find them fibroblast specifics. And that even goes to hematopoietic cells, that you have many of the hematopoietic markers you do find on melanoma cells. And this is one of the reasons why the field doesn't really have a good marker to target melanoma cells with monoclonal antibodies. There's no single monoclonal antibodies, and we and other people have tried over and over again to identify that. So far, we can live with this, but monoclonal antibody therapy in melanoma has had always a short-lived light, and then it darkened again, because the similarities of these epitopes with those on normal human cells were detrimental. So the final challenge that I just put in the room here is that melanoma cells can sit around. They can be dormant for years. And they can even have all the markers in the world of senescence, and they come out of it. So that's what one may call a senescence mimicry or senescence, pseudosenescence, and it can be quite confusing. And people have presented data on cancer-associated fibroblasts in melanoma, and then they have also sometimes had to concede that these indeed were not fibroblasts, but melanoma cells. So melanoma cells are plastic. That's basically we have to live with. They can change their phenotype, and they are a heterogeneous population. And if you clone cells into single cells, expand them, they would regain some or even most of their heterogeneity over time. And then the tumor is notoriously drug resistant because it has outstanding pumping system. Any drug that you give to the tumor cells, they're really good at pumping it out. They have a wonderful sort of stress response, autophagy system that helps them. It doesn't necessarily send them into apoptosis, but it is a supporting mechanism. So all these things together say, and that's why I'm not a primary an immunologist. My wife always was, but I tried to stay out of the field, but then I couldn't, because this cries for immunology to now interfere, since the signaling inhibitors almost invariably lead to relapse. There are some long-term responses, but overall, this has not been leading to cures. So our hope was and is that the immune therapy in one way or another can be combined with immunotherapy and then to eradicate a tumor. The melanoma as a field overall has been a front runner in studies on immunology. This is in part because historically the melanoma oncologists always had an interest in immunology, and the patients often have an active response and show a strong infiltration of immune cells, and thus many studies have been done in the past early on. When I started my career in the 70s and 80s, those studies were already gone. And at the end of in the 90s, the field was flat on the ground because by that time, nothing worked. It's absolutely, there was one phase 3 after another that got killed, and immune therapy did not work. This all changed a few years later, 10 years later, with the checkpoint inhibitors. And they are still riding high, and PD-1 as well as CTLA-4, now LAG-3 are 3 checkpoint inhibitors that really have an impact. They are similar to targeted therapy, that they provide a long-term response, but they also generally in 3/4 of the cases to 2/3 of the cases do not provide cures. So it appears that there is a 1/4 to 1/3 of patients that remain long-term stable and without evidence of disease. So, and it is, at the end, it's going to be a combination of immune therapy and targeted therapy. And we were particularly interested in also what these tumor infiltrating host cells, of course, the specific T cells, but then we were also interested in the macrophages, as many others, and the B cells. We had a study not long ago that actually B cells can be good for the tumor because it can provide the tumor with growth factors that they don't produce. In this case, it was IGF-1, and they are activated by tumor-associated FGF, and then they start to provide essential IGF-1. So those circuitries have always been an interest. We have done a few studies on suppressor cells, but many others have done so. So let me go through the model that was important for us, because we saw that without a combination with the help of the immune system, we cannot really bring the tumors down in long term. And we developed 2 models. I will only speak today about the first model, an HLA matched model, where we have the CD34 cells from the fetal liver, and as well as we then graft thymus pieces from the same donor. And these 2 pieces are then matched to the HLA-A allele of the melanoma, or the other way around. We pick that melanoma that matches to our donor. Here is a second model, and I'm fascinated by this model, and I'm fascinated, I have to admit, for 4+ years, and we are not there. So I will not concede that we failed. It is an incredibly challenging model. I will provide you an overview at the very end and tell you also where our challenges are, but it is a fascinating model, and I'm thrilled to be working on it. So we are continuing in this, even if we are not there yet. So let me talk about the what we call the fetal liver CD34 model. And I have to point out to Raj, who we all called Sham Somasundaram, who was leading this program. And unfortunately, a good year ago, he passed away, and it was really tragic for the laboratory, and we had to start all over again. And these were the colleagues who worked with him on the model. So we myeloablated. In this case, we used busulfan. Now we got a new irradiator, and we can do the irradiation. We graft the mice, we graft the thymus under the kidney capsule. And then I think this is critical. We stimulate with cytokines. And then we have the readout. So let me explain the cytokines, because this is, I think, critical. Here on the lower side, you see the red ones, what we call combo 1. These are the cytokines we overexpress in the mouse using an adenovirus 8 vector. The adenoviruses in general, as you may know, they have an affinity for going to the liver, so they settle in the liver, and they then integrate and produce these factors. Our experience was about 4 or 5 weeks. And the problem with associated adenovectors is you can't inject twice because there's a strong immune response. And then we have a second set of cytokines. There are some variations, minor variations, some one less, one more. That is what we call our combo 2. These genes we transfected with the gene gun technology. It's an intramuscular transfection, and actually the secretion of these growth factors is quite robust. We detected it between 3 and 4 weeks, and one probably could find it more. And then one may also not need it as longer. And then we looked for what came out. We did not study these precursor lesions too much, but then looked at, and those two we have not found. All the others we have found. So and you may have seen this study from the Flavel lab, who I still consider the premier lab in the field, who knocked out an enzyme in the liver of the mouse, and then they got red blood cells. They may even work on platelets, I don't know. For us, so we have not done much work with cord blood cells because we early on found much better results with the human fetal liver. I am not an expert on the cord blood and on the limitation also of this. We really like to have thymus cells in the same individual, and we could see that this thymus, the grafted thymus, the human autologous thymus, is infiltrated with lymphocytes. We can come back to it at the end, and I'll be happy to address some more detailed question. Now, these are the initial characterization and then of T cells and B cells, of CD4 and CD8. I think we got the whole variety there. And what was interesting was the sequence. There is a dynamics in it. So we found early on, after about 2 weeks, we found the monocytes or some DCs. Then the B cells were very high. And once the B cells went down, then the T cells came up. So we had some cases where the B cells stayed very high, and then the T cells didn't really come up. We in generally are using the mice if 20% of the PBMC in the mouse are human, and then there is a percentage of 8 to 10 of CD3 positive cells that represent CD4 and CD8 cells. And so those, and then I'll show you some of the pictures here on the CD4 and CD8 in the tumors, and the myeloid cells in the tumors. So we saw then in general that the tumors that again, they are HLA-A matched. When I should just throw in here a remark, and some of your experts will have heard several similar remarks, that when we started the project and I talked to my senior immunology colleagues at Wistar, they just told me, you're crazy, because if you come with this type of project to our study section, the people will kill you. And I have to say, the picture has changed. So that may have been still 5, 6, 7 years ago, but now I see still the classical mouse geneticist, immunologist working with humanized mice. So it has come a long way, and certainly in large part also through the major efforts by the Jackson colleagues. And so I congratulate you on your persistence of not listening to the hardcore immunologists. And so what we were surprised was the variety of these of the immune system. Here we saw then in the humanized mice, the TCR sequencing in this plane, meaning we get many different specificities of the T cell receptor. This was done by Jürgen Becker and his postdoc, and it shows that there's not just a few epitopes that are dominating the response, but it is quite widespread. And so we then started off with PD-1, and I think there we saw what we have expected. It worked some, but it didn't work completely. And that, of course, puzzled us. And we did then some RNA-seq and also some multiplex immunohistochemistry, CyTOF, to characterize what were sort of, why is there not a complete removal or regression of the tumor, this will be the major point here. So these are the CyTOF characterization of CD8 cells, of nestin, of HLA class, and then we could see that there was some regions, you see it in the upper right, regions of downregulation of class 1. Not being a hardcore immunologist, I wouldn't have seen it, but I credit my colleague Sham to this observation, which I find quite remarkable, that it is on a localized level. Yes, you find tumor suppressor cells, and then you find mast cells in there, and you find cytokines increased, And as you see here, in a number of different PDX treated with anti-PD-1 in the humanized mice, you find an increase in mast cells. So that was then a observation. And of course, we had not studied the mast cells before, but certainly, mast cells are a kitchen sink of cytokines and growth factors, and they have not gotten much attention in the scientific community. We then saw that in patient tumors, we see actually a very similar phenomenon, that there is a quite some strong, at least in some patients, strong immune infiltration of mast cells after anti-PD-1, and whether that was now much more after treatment or before, these are too few patients to make a good statistical comparison. But I think the overall, there are frequently mast cells, and they have not been on the radar screen by our oncologists. And then we looked at markers. It's difficult to find specific markers for mast cells. Mast cells are usually characterized by c-KIT expression, CD117. And so we used an old receptor kinase inhibitor, sunitinib, that targets c-KIT, but it also targets VEGF and many other kinases, receptor tyrosine kinase on human cells. And so we didn't like it too much. The reviewers didn't like it too much, but we didn't have a specific way of eliminating that. And but you see here, there's a remarkable synergy with those two strategies. If we eliminate the mast cells, we see then a much, much stronger response. And as you see also, the pure anti-PD-1, sometimes it has a strong effect, and other times it has in the model not much of an effect. And so we characterized this a little bit more. We then used also the classical c-KIT, that is inhibitor that is used for GIST tumors, and that's imatinib. And it worked in the same way. And it was actually, that's quite a, whereas one that has mostly only VEGF receptor inhibitor activity is less active than imatinib or sunitinib. So we then, of course, could show that the mast cells disappeared here, and that in the anti-PD-1 you have still plenty of mast cells. This is our current scheme. We have this intercellular interaction between the mast cells and also the activation of suppressor cells, the production of chemokines, and most important is the CCX cell 10, or IP gamma 10, I usually call it, that downregulates in a local level the class 1 expression on the melanoma cells. So this allows them then to escape from cytotoxic T cells. So that is the mechanism. We are convincing our clinical colleagues to develop a clinical trial. They don't like sunitinib because it has some toxicities. They no longer, the melanoma field was very disappointed with imatinib, even those few acral melanoma. There's a subgroup of tumors in Asians and in non-whites that have acral melanomas on the palms and soles. The success in treatment, they expressed c-KIT, and the success was not very encouraging. So that is, we are now using, trying out in the humanized mice another series of mast cell specific inhibitors. Those studies are just being initiated, and we don't have any experience. So let me briefly summarize here. Our job was more to do proof of principle, and we think we succeeded there. What we were very encouraged is the broad lineage specificity that we saw of the many different cell types. That PD-1 is can show an anti-tumor effect. Yes, and that we have the heterogeneity of the response. That most likely the tumor suppresses cells, like FOXP3 positive cells, are involved in the resistance, and that we can have a major impact if we combine immunotherapy with targeted therapy. That actually was our original goal, and I think that goal we showed in principle. Those clinical studies are still ongoing. They are still not easy because if you use only PD-1, it does have some toxicities, but if you use PD-1 plus anti-CTLA-4, it has some major grade 3 and 4 toxicities, which are really hard on the patient, and they may have to disrupt the treatment. So that's a hard drug to deal, a combination to deal with. And then the big question is, do you, and I have here some more of the future studies that are ongoing, is what you should do first. You do first targeted therapy to kill off the bulk of the tumor, and then immune therapy. And the answer is it doesn't work, because once you have used targeted therapy, immune therapy is less effective, unfortunately. So can we develop resistance models? And it's not clear. We think so because we have not worked yet with LAG-3, but plan to do so in the future. And CTLA-4 is decently well tolerated in the mice. Remember, it's the human anti-human CTLA-4. I've forgotten A there. Does the resistance lead to epigenetic and genetic changes? We don't know that yet. It is not clear. Once we have the models, it's easier to tell. We have, for targeted models, if we take a sensitive tumor, treat the tumor with a BRAF/MEK inhibitor, then after several months, those tumors, they will reemerge from a dormancy where you may have some tiny tumors left, and then they will grow. And so we can study those changes. And we hope, that is still our big hope, that the immunotherapy will kill off these drug-tolerant persister cells that we have in kinase inhibitor studies. And so the final question And let me give you the final slide there, experimental slide on our model, how we are seeing the future. And I have to admit, it's a tough future. So we make a PDX, and in melanoma, it's easy because you have a take rate of anywhere between 80 and 90%. And those that don't grow, there's usually a problem in your own laboratory, or there was something with the tumor. So, and maybe some accidents or anything like that. So we have our experience with C. bovis infection and the whole thing. So you then can grow fibroblasts from the patient, or you just isolate the PBMC, just 10 ml of blood is fine. You isolate those cells and use the 4 Yamanaka factors to induce iPS cells. This so far is routine, and thanks to Yamanaka, who has developed this about now 14 years ago, I believe, 14, 15 years ago. And then comes the big thing. We found a way of making CD34 cells. So these, we use the mouse. Remember that the iPS cell induces a teratoma. It's a benign tumor. It takes a little long, meaning 15 to 20 weeks. And then these tumors, if you co-inject some mouse mesenchymal cells, these are the OP9 cells, and use these developmental factors, you get a relatively high number of CD34 cells. High number, I would say you get about 1 million to 3 million per mouse. That's already a good start. And you know how few cells you can get from a cord blood. And then the big question is, can you then drive those? We had experienced to make melanocytes from iPS cells, so that's why we were optimistic. However, making hematopoietic cells is a different ballgame. It's about 10 times more difficult. And in melanocytes, we found a sledgehammer, and then everybody else was using that sledgehammer. It's actually a 4-wall ester that works perfectly, meaning a very strong driver of signaling, and then it worked. And that we have not found in the iPS cells. We are in the screening business. Screening is if you don't know exactly what will be the magic solution. So we have now screened about 100 transcription factors. We have also screened about 250 epigenetic modifiers. So, and we are making progress, but it is not a home run. And I will leave you with this, that it is going to be possible. And I think one lab shouldn't do it by itself. It should be a concerted effort. And I think it can be done. Meaning, in this case, we will have an autologous system where we have the PDX as well as the hematopoietic system from the same individual. That's ideal. You would say, why don't you use it from a patient? I think it is not practical. It is feasible that you get bone marrow from patient. I personally never wanted to go the route. I even found it unethical because the patients are often moribund, meaning sick, and then tapping their bone marrow is a hard job on these individuals. So we never wanted to do that. We think that this is, on a long run, the future. We have actually thymic progenitor cells. We have the 2 major cell types. We can make organoids of these thymic progenitor cells. And right now, we have them infiltrated with lymphocytes and see how we can modify the maturation of the T cells. So it will be, it's an interesting experiment. It has many different facets. And all I can say is we are not there, but I think we will be there. With this, let me thank you for your attention, and I'm happy now for questions.